வீடியோ மட்டும் அப்புறம் ஆன் பண்ணிக்கோங்க. ஒரு 10 minutes ஆகும்.
புரிஞ்சு பார்த்து பேசுறேன்.
எஸ் சார்.
அப்போ திருப்பிக்கலாம். இந்த பேக்ரவுண்ட்ல கரெக்டா
இந்த பக்கமா
கரெக்டா
பேஸ் மட்டும் தான் தெரியும்.
உங்களோட ஓகே பாருங்க, அப்படி தெரிஞ்ச ஓகே. இந்த பேக்ரவுண்ட்
இந்த இன்னைக்கு விட்டுருங்க சார், இன்னைக்கு வேண்டாம்.
வீடியோ
இதுதானே சார் அந்த நோட்ஸ்.
இது வச்சுக்கலாம், ஈஸியாக தான் இருக்கும்.
எழுதும்
எழுதி கண்டினியூ பண்ணிக்கலாம்ல. ஏன் உங்ககிட்ட புக் கூட இல்லை, ஸோ சொன்னாலும் தெரியாது. புக் வந்துருச்சான்னு பார்க்கலாம், வந்து அதுக்கேற்ற மாதிரி
ரீடிங் வந்து இது சார். எழுதும் போது இதை கிளிக் பண்ணி எடுத்துட்டு அதுக்கப்புறம் மேலே போகணும்னா இந்த ரீடிங் கிளிக் பண்ணுங்க. ஸ்டூடெண்ட் ஒருத்தன் வந்திருக்கான். ஓகே சார்.
Good evening, sir.
Yeah, good evening. How are you?
Fine, sir.
Good evening, sir.
Yes, good evening. How are you?
Yes, sir, I'm fine, sir.
Yeah, good. So, you got your books? School? You received
No,
your books?
sir.
No,
we,
sir.
We still do.
Fine,
Sir,
fine.
we are in our summer holidays, sir, so we won't get this for like now.
Okay, okay. Then we will have our free flow classes, right?
Yes, sir.
Yes, sir.
Okay, good. Then. So we'll start or wait for a few more minutes.
So we can start.
Hmm?
We can start.
You can able to see me?
Yes, sir.
Yes, sir. One more person has to come, sir.
Yes, sir.
Okay, okay. We'll wait for a few minutes and then we'll start.
Okay, sir.
Okay, right. So you remember the things which we discussed in the previous class? Have you done any assignment?
Yes, sir. I have done it, sir.
Yes,
Okay.
sir.
What assignment?
So you've given us 5 questions, sir.
Yes,
Yeah.
sir.
Easy? The questions are easy?
It was quite easy, sir.
Yes, sir.
Okay. Like all of the things which we discussed only, like
Yes.
was there in the assignment, right?
Yes, sir.
Yes, sir.
Yeah. What is the cell?
Sir, but actually I didn't mark down the first one and for marked down the answers for the first one and the second one. So I had to write it a little on my own.
Okay, okay. That's fine then. That's fine. Okay, fine.
Yes.
So maybe like if you would have referred your book, then that was easy for you to like connect the things which we discussed. Okay, like maybe because you may not have you're not having your book, so that's why maybe little difficult. Okay, good. So
Yes, sir.
shall we start the today's
Yes, sir.
discussion?
But one more person didn't come yet. It's
Okay.
I'll just call him, sir.
Yeah, just check.
Sir, did you have a share?
Yes.
Did you have a share?
Yeah, yeah. Yeah, you can't recognize that.
No, sir, the face is a bit different.
Yeah.
yes, sir.
You thought another person is sitting.
Yes, sir. Like
Yes,
first
sir.
when I joined, I actually thought some other.
Okay, okay. So you like the cell?
Yes, sir.
Yes, sir.
Okay. Fine. So let him come. And before
So
that,
he
let
came,
us
sir.
He came,
Yeah,
sir.
good. Yeah. Hi. So hope all of us
Karan,
are
you're
here.
muted.
Yeah.
Hello, sir.
Hi, hi.
Sir, my camera is not working for some reason.
That's fine. That's fine. That's fine. Okay.
Yes, sir. Thank you.
Yeah. Good. And if you cannot understand anything, you please let me know. Okay.
Yes, yes, sir.
Fine. Good. So, and in today's class, actually, just a minute. Okay. I just turned off my video just to make you like my writing clear. Okay. If you cannot understand anything, you please let me know. Okay. So in today's class, you all able to see my screen?
Yes, sir.
Yes, sir.
Yes, sir.
Yeah. We will be discussing about microscopes. Okay.
Yes, sir.
Yes, sir.
About in today's class, we are going to discuss about
Microscopes.
Microscopes.
microscope. Okay.
Yes, sir.
In microscope, the principle and applications. Okay, even you can say components.
Yes, sir.
Okay, the
Yes, sir.
principal components and applications of
Application.
microscope. See,
Yes.
what is the objective of knowing about microscopes?
So
So without microscopes, there's no like.
just to
Yeah.
observe
Yeah.
things that we cannot see with the naked eye.
Yeah.
Yes.
See, cells are microscopic.
Yes. Not visible to naked eye.
Yeah, not visible to naked eye. So you need a microscope to observe the cells, right? You need a
Yes, sir.
microscope to observe the cells. And one simple question.
Yes.
All the cells are microscopic?
So one cell, an egg is a cell that's not microscopic.
Yeah,
Yes, sir.
egg cells. Okay, egg cells of
Yes.
many organisms. Not all of the organisms egg cell, but many of the organisms egg cells maybe in a
Possible to see with the normal eyes.
yeah, with in a naked eye.
Yes.
Okay, few of
Yes,
the
sir.
egg cells can be seen with the naked eye. Even some neurons. Okay, in our body also we have some neurons which are very long.
Yes.
Okay, some there are
So
some
those are
cells which
visible
can
to
be-
our naked eye?
No.
Yeah.
So
Means
how, sir?
long neuron. Okay.
So
Means
I know that
they are-
elephants' neurons can be visible with naked eye, but I don't know that
see,
humans.
there is only one neuron in our body which possesses a
So it's like the thing that
long
goes from spinal
axon.
cord.
Axon. Okay, when it possesses a long axon, you know the basic structure of a neuron. It has
Yes, sir.
dendrites, cell body, and then the axon, long body,
Yes, sir.
with long tail-like
It
structure.
connects
and then-
the connects the nucleus head to the muscle cell.
Yes,
Neuron
yes.
head.
So some neuron can be seen. Okay,
Yes, sir.
means the thing which we should know is some cells can be seen without naked eye, but most of the cells are microscopic. Okay, most of the cells
Yes,
are
sir.
microscopic except a few cells. Just keep that in mind. Okay, fine.
Yes, sir.
Okay, to understand the discovery of microscope. Okay, about the discovery of microscope. Who discovered microscope?
Robert Hooke.
Robert Hooke.
Okay, see, actually, many people comes into the picture, and first lens makers basically like Zacharias Janssen. Okay, he is the first person, but maybe
Sir,
in your
can you
higher class,
spell, sir?
yeah. Okay, Jason Zacharias.
Sir, is there a date? Oh, you don't need to remember.
Actually, just a minute, 1 second.
Yes, sir.
So this person actually, even in 1500s, okay, father and son, okay, like in 1500s, they just used lenses. Okay, Janssen basically, I think Janssen in 1500s they used lenses to observe microscopic substances, small substances. Okay,
Yes, sir.
first important thing, and then coming to the cell biology. Okay, coming to the cells, like as you said, the first person is Robert Hooke, basically. Okay, he observed dead cell. Okay,
Sir,
Hooke,
but in the book it says Anthony van Leeuwen.
Actually, Anton van Leeuwenhoek designed the microscope. That's about how microscopes are designed. Microscopes are designed for various
Wait.
purposes.
Didn't
Okay.
our teacher say Robert Hooke was the first person to observe a cell under the microscope?
See,
No,
that's
he
what.
developed.
See, listen. Microscopes like like Anton van Leeuwenhoek was constructing microscopes for his own purposes.
Oh.
Okay, almost 400, many types of microscopes. He tried a different combination of lenses just to magnify simple substances. Right?
Yes, sir.
He connected two things. One is microscope and then the cells. Okay.
Yes.
One is
So
microscope
can
and
you
then
just write his name?
just a minute.
Anthony van Leeuwenhoek.
Yeah.
You don't have the copy of the second chapter, does uncle?
No, I don't
You
have.
can download it from internet.
Yes, it's in
Okay.
studiestoday.com. That's where I do.
Studies today.
What's the second chapter?
Actually,
Cell.
this is the second. Unit 1, Basic Biology, Chapter 2.
It's a cell.
So
Yeah.
cell is the first chapter.
No, it's the second. Ma'am, it's a
Second.
cell.
Oh, yeah.
Yes, sir.
Can you see?
Yes, sir. So you're selling Anthony van Leeuwen who didn't invent it, just
He was
Jason
constructing
made it.
microscope, simple microscope he was actually constructing. Okay.
So he was making Jason's microscope
Ah.
better and trying different
Yeah,
lens.
he is the like they were initially talking about, means they were working on designing microscope with lenses to magnify substances. Lens makers, magnifying substances, no way connected to biology.
Yes, sir.
Can you understand?
Yes, sir.
Yes,
So
I-
Jason accidentally invented the microscope.
Yeah. Means the purpose changed. Initially, like since they wanted to make some materials with more cautiousness. Okay.
Yes.
And the designing, all these things basically. And then the purpose changed. They because cells itself discovered after the discovery of microscope only. Okay.
Oh,
First they
Yes, sir.
discovered microscopes and then only the cells were discovered. Is it clear? So first
Yes.
microscope comes and then only the other application. Actually microscopes were not designed to observe cells. You know that, right? They are not designed to observe cells. Like they just
Sir,
meant
which year is Aristotle, sir?
Aristotle is 300 BCE, right? Aristotle, the
Sir,
father
but
of
yes.
science.
didn't he also talk about
That's what.
cells?
See, listen, many people comes into that action. Okay. 1 minute, I will give you all those data also. The history of microscope. Just a minute. I
Yes,
will
sir.
give you that. Just okay. And please note down certain things. Okay. And we are now actually starting, since you are asked for that, we are starting with what? The history of microscopes. Just a minute. Oh, for just a minute, my
Yes, sir.
please wait a minute. My screen got locked. Let me ask them to switch it on. Just a minute.
Yes, sir.
Okay. Hello, students. Can you able to see the screen now?
No, sir. There is no. You
No,
didn't
sir,
show.
you didn't. Yeah, you didn't share your screen, sir.
Just a minute. Now, Hello, students. Can you see the screen now?
Yes, sir.
Yes, sir.
Yeah, the
Yes,
white
yes, sir.
screen.
Yes, sir. We can see, sir.
Okay, fine. So now, yeah, see, there are the history. We are going to discuss about the history of the discovery of microscope. Okay, history of microscope.
Micros.
Okay, history and discovery of microscope. So the first thing is actually like Hans Lippershey, Lippershey.
So who
And
is this, sir?
I'll tell you. And
Yes.
Zacharias Janssen. Okay,
Yes, sir.
okay. In
Yes, sir.
yeah, they are actually, he is the father, he is the son. Okay, in
Yes, sir.
1500, actually they are the lens makers, spectacle makers basically. In
Sir,
1500,
can
Who's
you,
the first person?
sir, can you
yeah,
spell the first word again?
yeah, Hans, Hans, Hans Lippershey, Lippershey, Hans Lippershey,
Litter.
Lipper,
Then
Shey.
yes.
And
Second one?
Zacharias, Zacharias, Zacharias Janssen, Janssen.
Yes.
So in 1590s, okay, they are the spectacle makers. They only used different lenses, and it was very simple, like which could able to magnify things. Okay, the first device which can magnify things we call now as microscope. Okay, so this was the first important discovery. Okay,
Yes,
and
sir.
next, Galileo.
Yes, sir.
Okay, yeah, Galileo, right?
Yes, sir.
Just a minute,
Yes, sir.
I'll write it. Galileo, can you understand
Yes,
the
sir.
spelling? Galileo, he invented his microscope in, actually he called it as what? Little eye. Okay, when in 1609, okay, in 1609, Galileo invented a microscope which is called little eye. Okay, very simple design. And then the third person only, Robert Hooke. Okay, Robert Hooke. Okay, in 1665. Connected with the cell biology. Okay,
Yes, sir.
he observed, listen, see, these two people, Robert Hooke and Anton von Leeuwenhoek, he observed dead cells.
So Galileo
Dead
made
cells
a
of,
microscope which Robert Hooke used.
no. Robert Hooke designed his own microscope. Okay,
Yes.
Robert Hooke designed his own microscope and observed. He is the one who reported about
The
the
cells.
basic arrangement of cells.
Yes.
Okay, about cells. Okay, dead cells he observed.
So
And
Galileo
like this,
is the one,
basically
like
microscope,
Galileo is
like
the
this,
one
like
who
this.
reported. The
No,
microscope.
Robert
Galileo.
Hooke is the one who reported.
Galileo used microscopes which was used to observe things, magnify things, just the magnification.
So
Okay.
he doesn't have a
He didn't,
actual,
yeah.
he doesn't have any
Galileo
motive.
did not observe any of the cells. Okay. First time cells were observed by Robert Hooke in 1665. Okay.
Yes,
And
sir.
see, listen, in history, what we will consider, maybe other person would have also seen, but if it is reported and if it is recorded, that alone is considered in the history. See, Robert Hooke in his publications, Micrographia, his publication is called what? Micrographia.
Graphia.
Okay.
So when
He
he
is
looked
the
at
one
these
who,
cells, he named them micrographia.
no. This is the article. You can say it is a magazine, research article. Okay. Micrographia is the research article. See, he saw, if you know the plant cells will have cell wall, right?
Yes, sir.
What will happen to the plant cell if it is dead?
A dead plant cell.
Yeah, it stays as it. See, look at that. Robert Hooke observed plant cells, group of plant cells, but all were dead. Only what is there, he can see. Cell wall.
Cell wall.
So, can you say this is like in those days, the monks used to stay in a little room which is called cell. Cell means a little room. Cell means
A prison-like thing.
Yes, like cell, a little room.
It's
Can you
called
say a
a
little
jail.
room?
It's also called a jail.
Yes.
Little room where not just the criminals, even the monks, they live very simple life, right?
Yes,
Very,
sir.
very simple life. So he saw such kind of arrangement, small room, many rooms you can say. That's why he coined what? Cell. It looks like a room, small room, but all were dead. Can you understand?
Yes,
Yes,
sir.
sir.
Fine. Look at the year he published. That's why we are considering him first. Okay.
Yes, sir.
Next, even before that, in your book, you can see when Anton von Leeuwenhoek discovered microscope.
Yes. Yes, sir.
Hmm?
Yes, sir.
When Anton von Leeuwenhoek discovered his microscope.
We have to tell
Yeah, I'm asking you.
the year, sir.
Yeah. Actually, in 1630, his period is
1632 to 1723.
Yeah. So,
Are you talking?
Yeah. Can you hear me?
Now I can hear you, sir. Now
Yeah. Next person is Anthony Van. Yeah. Leuven. Can you understand my spelling?
Yes, sir. Yes, sir.
Leu. Leuven. Hook. Okay. Anton van Leeuwenhoek. In your book, you can see 1632 to 1723.
Yes, sir.
Okay. So he designed his own microscope. Okay. And his microscope is called actually what? Simple microscope. Okay. His microscope is called what?
Simple microscope.
Microscope. Okay.
Yes, sir.
Robert Hooke microscope is called what?
Simple microscope.
Robert Hooke microscope is called what?
Compound microscope.
Compound microscope. Okay.
Yes, sir.
Now we actually call it as compound microscope because it had a greater resolution.
Magnification.
Yeah.
So sorry,
Resolution and magnification. The principles we will see. Okay.
Yes, sir.
Okay.
Yes.
See, what is the significance of Anton von Leeuwenhoek's contribution is, he is the one who observed live cells, okay,
Yes, sir.
live cells, and bacterial cells. He saw bacterial cells, and
Yes,
he
sir.
saw, and then
So,
he saw,
so he saw a prokaryotic cell even before a
yeah,
eukaryotic.
he saw the shapes and hue. He he drew all the diagrams and presented, and later only we call them as what? This is a bacterial cell. He just presented the images.
So
right?
why does everyone tell
So
Robert to put
actually,
the first question?
I'm not gonna lie, but
Ah.
like the diagrams given in the book are the observations drawn by the scientists.
Yeah,
But
you can say in
yeah,
Robert
it is in a simple microscope. So in our school we have a simple microscope, and when we look into it, we see a plant cell. And when we see a plant cell, it is very less identical to the one that is drawn in the board.
Ours is
Yeah.
better version of the simple microscope, but it's still simple microscope.
Yeah, but
Yeah.
like our version, like our microscope, which is a better version of a simple version, is which is actually showing a very is showing an image which is very hardly very hardly identical to the one drawn in the textbook and board.
And
Then
that's because, see, listen, that's because using microscope is a great skill. Okay.
I think
So,
they saw different stuff.
yeah, like the kind of lenses, how we are going to focus things. That is what matters. See, there are different adjustments, fine adjustment, coarse adjustment, different components we have. If everything sets, and then you can able to observe things clearly. Okay.
yes, sir.
Yes, sir.
Yeah, you have to know about all those things. Then only it is easy for you to, if you develop the skill, then it is very easy for you to understand.
Yes,
Yes,
Means you
sir.
can observe even more clear things. Okay.
Yes, sir.
We
Yes,
will-
sir.
Actually, we may have some kind of assignments like usage of microscopes. Let me discuss with sir one day. Like we will plan anyway. Like I have to discuss with that. You should be given an hands-on experience on using microscope. We'll try.
Yes, sir. It
Yes,
seems
sir.
that like few Saturdays, Sundays,
Yeah.
once in a month, we come to Velachery learning room.
Yeah.
And
Somehow we will definitely plan for that. Okay.
yes, sir.
Yes.
So
Yes,
can
sir.
you tell the spelling of Antonio van Leo?
Yeah, Anthony,
He wrote here.
Antony, Anthony Van, 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 Leeuwenhoek, Leeuwen, Leeuwenhoek, Hoek.
Okay,
Okay.
sir.
And around 1670s only he reported. Okay.
Yes, sir.
Yes, sir.
Anton Von, and he saw, see, listen, he draw the structures of what eukaryotic,
The plant cell.
The actual
bacterial,
complete
plant
cell.
cell, even animal cells also. Okay.
Yes, sir.
Animal cells.
They didn't
Or
find
sperm
human cells, sir.
especially, yeah, the sperm cells, human sperm cell structure. He is the one who first published.
Yes, sir.
Okay. All type of cells, bacterial cell, plant cells, animal cells, he actually observed, even blood cells.
So how do you get a sperm cell, sir?
From seminal fluid.
Like you have to take it out of the
Yeah.
male's body, like.
Yeah, in male's body, after ejaculation, the sample has to be observed. Okay, what is there in the sperm cell? Means seminal fluid. Okay,
Yes,
like
sir.
that only people observed. He saw in blood is there. See, listen, people were wondering, what is blood? What is there in the blood? What is semen? How baby is formed? There are different theories are involved. Something called, some people will say panspermia. What is called panspermia? In the sperm cells, spermist, ovist. One concept was there, something called what? In those days, people called spermist, ovist. What are called spermist and ovist? They were arguing that miniature exists in the sperm cell. See, this is the sperm cell. Okay, this is the sperm cell. Head is there, and then tail. People thought that a miniature, a baby is there in the sperm cell. Then only if fertilization occurs, egg is fertilized. This is what their idea. They supported this. And people were telling ovum. Ovum means what? Egg cell. They were telling no, miniature exists in the ovum. And if sperm cell fertilizes, it gets activated. Baby is developing. Okay. So
Yes,
they analyzed sperminal fluid and vaginal fluid. Basically, they analyzed and came to know the structure of sperm and egg cell. Okay. And he is the one who actually presented the structure of red blood cell, sperm cell, bacterial cell, lot of cells he actually observed.
So the problem for the new upcoming scientists is in the olden days there was a lot of things to discover and invent. Now there's nothing left.
Yeah. Now
there's so much in the ocean.
Yeah. See, now the properties need to be discovered, studied, and connecting with our daily day-to-day life. All these things are the greatest challenges. Right?
Yes, sir.
Yeah, fine. Okay.
Yes, sir.
Yes, sir.
Fine. Next person is actually coming to the history. Malfigi. Okay, what is his name?
Malfigi.
Person, I think. How many persons we discovered? 3 or 4?
Yes, sir.
Fifth one. Okay. Actually, he is called what? The
So can you spell him?
I'll tell you. I'll write. Don't worry. Can you see?
Yes, sir.
Yes, sir.
Malfigi. Okay. And then other person along with him. The other person is actually called what? Nehemiah Grove.
So these are like partners in the
actually in late 1600s, means late 1690s basically. Okay. And then another person is actually called what? Together basically Nehemiah.
Nehemiah.
Nehemiah Grew. G-R-E-W. Grew. In early 70s. Okay.
Grew, that you're telling he grew or it's his name?
It is his name. Nehemiah Grew.
Oh, okay.
Early 1700s.
So if he grew in early 17s, how can he be a partner with?
No, he did not grew. It's his name.
That is his name. Nehemiah
Oh,
Grew.
sorry.
Yeah, like see, they studied and they published what? Detailed structure of plant cells.
Yes, sir.
Okay, detailed structure.
They studied or they discovered, sir?
Yeah, means all
they
plant
discovered
related
and
components.
published and studied.
Yeah, both. They studied and they published actually. They recorded, they reported. Okay?
Yes, sir.
Fine. Okay. So, and then the other person actually, another important, significant person in the history is actually called what? Robert Brown.
Robert Brown.
Yes, sir.
Yeah. We are just considering the most significant discoveries. Okay, in 1800s.
Yes, sir.
Okay.
So that means there are more than there are like So many discoveries.
Yeah. So many discoveries. I'm just listing very significant discoveries.
Robert found an nucleotic, like the head of the thing, sir.
Yeah, nucleus. You know nucleus.
Yes,
a
sir.
cell will have prokaryotic cell. A
Yes.
cell will have an organelle called a nucleus.
Sir, but only eukaryotic cells have it. So
Only
prokaryotic
eukaryotic cell will
don't
possess
have.
nucleus.
Yeah.
And later only we came to know that not all of the cells possess nucleus. Only eukaryotic cells possess nucleus. We came to that later part only.
Yes, sir.
Okay.
Yes, sir.
Is it clear? So
Yes, sir.
Robert Brown. Okay.
Yes, sir.
So and look at the timeline. Can you see the timeline? First, understand the lens makers Hans Lippershey, Zacharias Janssen discovered a device which can magnify things. Okay.
For the help
And
of
then,
easier things.
Yeah. And then Galileo, he discovered a microscope. He just magnified things and observed all
Yes.
our non-living components
He
only.
observed.
General, all substances,
like normal
general
helpful
substances,
things.
not a
like,
living cell, but distant substances. He looked into sky like that, mountain from one place to other place, far away places.
How can he look into it?
Just if you get
But it's a microscope,
binocular,
right, sir?
see, listen. If you
almost like a telescope.
yeah, telescope. If you have a
He
telescope,
thought it was a telescope, sir.
yeah, telescope like thing only. Okay, just he observed space, distance, substances, materials present far away.
Sir, I know that Galileo actually was the creator of it, of the telescope.
Yeah, that's what. Similarly, like that.
Yes,
Okay,
sir.
and then how telescope works, magnifies. See, in distance it is a small point. When you look into the telescope, the small point is magnified and you can see that. The principle is connected with that, right? That's what. Okay.
Yeah. Yes,
Yeah.
sir.
And then these people, most significant people are Robert Hooke and Anton von Leeuwenhoek. Okay, Robert Hooke discovered dead cells, plant cells, and he called them as cell. Okay, just because of the appearance of cell wall.
Yes,
And
sir.
then Anton von Leeuwenhoek, who observed live cells, and he presented the structures also: bacterial cell, plant cell, animal cell, sperm cell, blood cell. A lot of cells he actually presented. Okay,
Yes,
the basic structure we actually presented. Okay,
Yes,
so
sir.
like that, the history after as the specifications and the advancement in the discovery of microscopes, we started understanding the cells clearly. Okay.
Yes, sir.
Just because of microscope, we started understanding the cells clearly. So these people were the significant people. Okay.
Yes,
Now,
sir.
why I stopped the history here in the sense, from here what actually starts cell theory. Okay.
Yes.
Some here cell theory starts.
What
And
about the electron?
Before, yeah. Before that, listen to me. Before starting to the cell theory, understand two things. Microscopes or the components. What are the components of the microscope?
The nose piece, eyepiece.
Yeah. Components of microscope and its principle.
High power lens.
Yeah, you have lenses. Correct?
Yes, sir.
You have lenses. Main thing is actually like
The
you
lens.
have different kind of lenses. Two kind of lenses basically.
Sir,
What are
I
the
know
High
that-
power, low powers.
in the sense eyepiece, nose piece.
Oh.
In simple, we call it as what?
Eyepiece, nose
piece.
piece.
Okay, lenses basically. Ocular lens and objective lens. Okay.
Yes, sir. In
Okay,
objective lens
lens.
only, it's high power, low powers.
Yeah, we will come to that basically. Lens, and then you have knobs, adjustment knobs. You have in your book. You can see the
Yes,
arm,
sir.
the stage.
Fine adjustment.
Yeah, coarse adjustment, fine adjustment norms.
Yes, sir.
Okay. And then the illuminator called light source.
The mirror.
Yeah,
The,
light
the
microscope.
concave
Yeah.
mirror at the
See,
bottom.
yeah, light microscope in the sense you use like electric power.
There is
And
no concave
simple.
mirror at the bottom. It's a plain
That is,
mirror.
yeah, plain mirror
So
which
there
can
is
process light.
there's a
See,
concave mirror, right?
No, it's
that's
a
what. See,
plain mirror.
listen. Yeah. See, listen. You can see two type of microscopes. One is called simple, and another one is called light microscope. Simple microscope and light microscope. What is the difference between simple and microscope? In simple microscope, natural light source. For that only you have in the bottom you can have a mirror. Okay.
Yes, sir.
Mirror. But in
Oh,
light
so
microscope,
that's why
electric
they
switch
keep
will
the
be there.
thing.
Okay.
Oh.
Electricity. Okay. Switch
Artificial
will be there.
light, like a bulb.
Yeah,
or
light source. Okay. And it is condensed also. Means in natural light, you cannot condense the light. As it comes, you will be using it. But in here, you who use a condenser will process your light.
Yes.
Okay. Means the thick light, like this, the light travels and it should be processed to one particular point.
A short
Okay.
beam.
Not
of light.
scattered. To increase the clarity, it is just the light, which is however it is coming, it is like this focused to one particular point.
So
Okay.
that's why in our schools and all they keep it next to a window. So
Yeah.
Yeah.
light
It should get the natural light. See, there we use basically simple microscope. Only
Yes, sir.
natural light source you use to magnify, means to observe things.
Yes, sir.
But in light
So-
microscope, you need electricity. Okay. And the kind of lens also. If a 10x means what? 10 times
10 times
magnification.
magnification.
Yeah. 100x means 100
Yeah,
time
100 times
magnification. See,
magnification.
there are two type of lenses we saw, right? One is
Yes.
ocular lens, eyepiece you can say in simple,
Ocular.
eyepiece, and then objective lens you can say. See,
Yes,
if it is
sir.
Ocular and objective.
yeah, ocular is eyepiece, eye.
The eyepiece and the nose
Objective.
piece, sir.
Yeah, objective is nose piece.
Oh,
Near the
okay.
object you
Yes.
have, right? So
Yes,
this
sir.
is, for example, this is 10x. In ocular 10x, in objective 10x. What is the magnification? How many times the image is
20x, sir.
magnified? Huh?
20x.
Yes, 20, 10 into 10, no, 100.
Oh, 100x.
100x. 100 times your image is magnified.
Yes.
Sorry, I
So
was
if
muted.
I have
So,
20 and 20, it'll be 200x.
yes.
No,
No,
wait.
right, it'll be
400.
400.
Sorry,
20 into-
sorry.
Yeah.
You failed
Okay.
in first
Or
grade.
if I have
Something.
20x in the
Ah.
eyepiece and 10x in the nose piece, it will
Yes,
be
yes,
200x.
So, what is the purpose of this lenses?
Sir, to
That is called, we call it as what? Principle of microscopy. Just understand what are the principles of microscopy.
yes, sir.
So microscopy.
Microscopies.
Yeah, microscopy is the field, microscope is the device.
Okay.
Principle of microscope or microscopy. Okay. See, two important principles are magnification. Can you understand this?
Yes, sir.
Magnification and resolution. Okay.
The resolution is in the sense the
Magnification is-
clarity, sir.
Magnification is
Uh,
enlargement of the size. Resolution is,
the clarity, sir.
yeah,
Yes.
clarity. Okay. Okay. The same principle is used in your mobile camera, magnification and resolution. But in microscope, magnification is given more importance.
Yes.
But in your mobile
Mobile
camera,
phone.
like the
Clarity.
resolution,
The resolution is more.
yeah, important feature in your mobile camera. Okay.
Yes, sir.
Fine. So what is magnification? Enlargement. Okay.
Yes.
What is resolution? Clarity.
Clarity.
Between two nearby points, more clarity should
Yes,
be there.
sir.
Okay. So for example, let me ask you some simple question. Say, if the eyepiece lens is, okay, your
Yes.
eyepiece lens is 10x. I can say eyepiece lens 10x.
Eyepiece.
Yes, sir.
Objective lens or no space lens is 4x.
Sir, 40x.
Yeah, 40x.
40.
In the sense, 40 times your image is magnified. Okay, mostly eyepiece lens will come 10x, 15x, 20x. It is fixed. Okay.
Yes, sir.
And depends upon the kind of
So, but we don't have a nose
microscope.
piece in our school.
No, we do. Ma'am,
that
just
only,
don't
it is actually directly connected. Actually, that is there nearby your object. You means in your school you cannot adjust it. It is fixed.
No, sir, there is, but ma'am doesn't adjust it for us.
Means
There
you
is
have to,
3 more lenses.
in your simple microscope, you have to removable lenses are there. You have to remove and fix new one. Remove and fix new one. The no space lenses are not fixed.
Like this.
Okay,
Not
they're
it has to be
switchable.
removed. It is fixed actually. Yeah, it is fixed. But you can screw type no space lenses are there. You have to screw it and open and then add a new lens. But
Yes,
in other light microscope and all, the no space itself you can see. For
rotate
example,
it.
yeah,
It's like it can rotate
yeah,
around
10x,
the body to it.
Yes,
Can
sir.
40x, and then you can even see 100x.
So
Also,
in
you
the
can
light
see,
microscope, there will be 3 scopes, each with
yeah,
different magnification.
10x, 40x. In a light microscope, the no space lenses are 4x, 10x, 40x, 100x. Can you understand?
The eyepiece is 10x, 15x, 20x in a
no, mostly any one will be there. Either 10x or 15x or 20x. And
so, but the eyepiece
preferably
is
10x.
removable and we kept in
Yeah,
a
I can take it like
Removable,
this.
but removable, but always they provide 10x only. Okay.
Yes, sir.
So, but
Mostly 10.
15 and 20x is also fittable for
The resolution will be
Depends. Yeah,
lower.
resolution will be affected. 10x is good actually. Okay, in light microscope, in lab purpose, 10x is good. 10 into 40.
So
10
then
into
an
100,
electron
times.
microscope, how much
That's
does
different.
See,
That's it's
only
beam of light,
the light
beam of
microscope
electrons.
maximum 1000 times. Electron microscope 1 lakh times.
Yes, sir.
Okay.
So it uses beams of electrons to hit the
No light is used. Electron beams alone are used. Okay. 1 lakh times.
Yes,
Is
sir.
it clear?
Yes,
so,
sir.
So,
but how is it possible, sir?
It is.
Yeah, that is the device. That is the device. That's
so
the principle
it's
of
mostly used to see microatomic.
like very small
atoms
particles, detailed structure of substances. Okay.
Yes, sir.
Fine. So, understanding cells, you should understand about the principles of microscope. Cells are microscopic. So, if you understand how microscope works, what are all the different components of microscopes, types of microscopes, then only you can understand when study the cells in detail. Sir. Okay, fine.
Yes, sir.
So, one more simple topic we will discuss and then we can go. That is called what? Cell theory. What is called cell theory?
The law which the cell is in.
Characteristics of a cell. The law which tells about the
yeah. Cell theory talks about the commonality of the cells. Okay, see, listen. Again, there are 3 different people contributed to propose cell theory.
Yes,
one is
sir.
called what? In 1838,
Matthias Kilden.
Yeah, a botanist. Okay.
So what's a botanist?
Botany,
the guy
plant,
who, the botany,
plant
the study of
botany,
plants.
plant biologist. Okay. Matthias
Sleeden.
Yeah, Matthias Sleeden. Matthias Sleeden.
So
he
who
proposed that he's the one, I'll tell you, all parts of the plants. He's the botanist. Okay, he told that all parts of the plants are made up of cells. Okay, all plant parts, like the leaf, the stem, the root, whatever, all plant parts are made up of cell. Okay. And the other person, in 1839. He is a zoologist. Okay, he is a zoologist. Means what?
Study of animals or like
Study of animals.
speaking to animals.
His name is what?
Study of
Theodore?
animals.
Yeah.
I'm talking about Juma. Sorry, sir.
Huh?
Nothing.
Theodore Schwann. Okay, he told all parts of animals are made up of cells.
So instead of this, they can just say all living organisms are made up of cells.
Together. That is cell theory. Wonderful. One person is a botanist. He observed all the plant parts and said, and then another person is zoologist.
So
all
they
animal
both
parts
combined
are
it
made
to
up
form
of
the
cells.
cell theory.
Together they proposed what?
The cell theory.
Cell
So
theory.
plant cells and animal cells are different from each
That's
other.
what,
Right?
but they're both cells, right?
That's what. Different only. Structure is different only. But cells only the common unity. Yeah.
Yes.
That's right. So together they told what?
All
Cell
organisms.
is the small, the cell theory. Three major points together. Okay.
Yes.
First postulates. You can see the cell theory or the first postulates.
The
cell
smallest
is the
unit.
smallest unit of structure of all living organism. What is it?
It's the smallest unit.
Yeah, is the smallest
unit
of life.
Of structure of all life.
Unit of structure of
all living things.
Yeah. Structure of all living things. Can you understand? Means in simple, all living organisms are made up of cell. Because of that only, like they told, the second postulates it's connected to that. The cell is the unit of, since all organisms are made up of cell, the cell is the unit of what?
Life.
Yeah, is the
unit of
function of all living things.
Yeah. Function of all living things.
Third one is all cells.
All cells arise from pre-existing cells.
See, that is postulated by what?
The study of cells.
No, the other person actually.
Rudolf
Virchow.
His name is what actually?
Rudolf Virchow.
Yeah, Rudolf Virchow. Okay, in 1858. Okay, the third postulates come in the year what?
1858.
1858.
Yeah, Rudolf. Okay.
So why do these people have? Are they like
in the olden ages? These were customs.
Yeah.
No, like all three of them are Germans.
1858.
Sir,
okay.
All three of them are Germans.
No, the first two are Germans. The Rudolf Virchow, we don't know.
German botanist, basically. Second also German botanist.
Okay. German zoologist.
Okay, fine. So
Rudolf
Virchow is omnis cellula. See, there is a statement called what? Omnis cellula is cellula. Fine, that you will come to later.
What is that,
but anyway, what says cells arise
from pre-existing?
Pre-existing. Only cell can be formed from another living cell.
Yes.
Okay. Pre-existing cell.
So in that sense, if like plant cell is made up of pre-existing cells, and there's a nucleus.
See, plant cell can make another plant cell. Animal cell can make another animal cell only. So, like as the father, so the son.
So, but a neuron cell cannot make a neuron cell.
Yeah. That is something called cell potential. That is a differentiated cell. But it came from a neuron stem cell. Neuron.
Cell theory is the theory that applies for all the cells, right, sir?
What?
Cell theory is the theory that applies.
That is why, see, listen, that is why we call cell theory as commonality. What is called commonality?
It's a similarity between every
commonality
cell.
of all cells. Okay, all living organisms. Okay, all living cells comes under this category. Okay, commonality of cells.
So they either have to have any of the three, or they can even have all three.
They should have all three.
Yeah, they must. They have actually. Okay, you cannot create a cell in your lab now. Can you create a cell?
No, sir.
Neuron
We need other cells to make
do
this.
not arise from
neurons.
pre-exist.
See, listen. Neurons cannot differentiate, but neuron differentiated from
Neuron cell.
neural stem cell. Okay. Neurons are differentiated cell, specialized cell.
So neuron cells were formed by neural stem cells.
Yes, neural stem cells. They are formed when you are in your, they started its generation when you are in your mother's womb. There itself neural tissue development started. And then all in your adult body, all the neurons actually differentiated.
Yes.
Only
So
the
it's like,
differentiated. See, not just neurons, pass, listen. Even your heart cell also cannot divide again, cannot differentiate again. Once it is formed, that's it. But your skin cells and all can differentiate. Okay.
yes, sir.
Yes.
Okay.
They die and they'll regrow.
It's like once they are created, they cannot. The neurons can't create itself again.
Yes. They lose its division potentials. Okay.
After a specific number of divisions, right?
Of course.
Sir, is it because they don't have chromatin fibers in their nucleus?
What?
Like, is it because
no, the nucleus can't divide more?
See, there are 3 types of cells in our body. Listen. Some cells can retain its division potentials. Okay, some cells can retain its division potentials. For example, you have hematopoietic stem cells, blood forming cells. Always new blood cells are formed in your body.
Yes.
Okay, and some cells only if necessary they divide. They have the regeneration potential. For example, you have your osteocytes, bone cells. They will divide. If you get bone injury, then only it divides. Otherwise, it retains, but it will not. Just like that, bone cannot grow just like that. Okay. If necessary, they divide. But neurons, cardiomyocytes, these 2 cells are well differentiated cells. Once differentiated, they will not divide again. They undergo cell cycle arrest. Something called later classes you will be studying about cell cycle. Every cell will have a cycle. They will be born and they will live, they will function, and then they will die. Like us, look at us.
But how is it a cycle if they die and then it ends?
Yeah, the old cells are replaced. New cells are formed. They disintegrated. They are disintegrated.
Oh.
Okay.
Okay.
Yeah, that's it. So we will be discussing all those things. So some cells can always differentiate. Some cells only necessary can differentiate, like bone cells. Neuron, cardiomyocytes, actually they do not differentiate at all. Okay.
Yes, sir.
Means cells have different properties. Okay, cells have different property. Okay.
Yes,
like
sir.
Means they are the structural unit, they are the functional unit, and they have a cycle. Means they are born, they live, they perform, and they die also. Okay. And then one thing they will say, life begins with
a cell.
Yes, life begins with cells. All the living organisms start their life with cells only. Okay.
Yes, sir.
Hence they are the basic unit of life. So we have to study about cells to understand the life. Okay.
Yes, sir.
Okay. How many cells we have in our body? You know,
more than 100 million
trillion cells. Yeah, 100 trillion cells we have in our body. It's very good.
In the book, it's given 1,000 million cells.
Million and million, 1,000 million and million means 100 trillion cells. Okay, lot of cells it means. Okay.
Yes, sir.
Yeah. How many nerve cells they are telling? The tidbit information in your book. 10,000 million nerve cells in brain cortex.
Yes.
Okay. Yeah. And then 5 to 6 million red blood cells in
A
1
body.
microliter of blood. Okay.
Yes.
1 microliter. So 5 million cells.
7,000 white blood cells per cubic meter of blood. So
It
isn't
means-
that very less?
It means what? See, very, very less. Red blood cells are present in millions.
It must be
White blood
cubic
cells
centimeter. No way it's
means
cubic meter.
millimeter. Yeah, millimeter cube. Okay, it is millimeter cube, means 1 microliter. Okay, what is ml? 1000
Some
ml is equal
milliliter.
to 1 liter. See, listen, milliliter. 1000 milliliter equal to 1 liter.
Yes, sir.
Okay.
100
Like that?
milliliter equals 1 microliter.
Means upon
No,
1000
100
See,
milliliter is
listen,
a centiliter.
1 in
The
1000.
liter.
Sir, it is not cubic milliliter, it is millimeter.
Millimeter cube. See, listen, that is the area. Millimeter cube. Also called microliter.
So in a
1
specific
microliter means
amount
what?
of liquid, there's this
See?
many red blood
Yeah,
cells.
yeah. Means why they are telling millimeter cube in the sense, we use a device called hemocytometer. Okay, we use a device called hemocytometer. In that, it holds 1 microliter of blood. Okay, it holds 1 microliter of blood. So we call it as either 1 microliter of blood or in 1 millimeter cube. About
Yes,
the
sir.
volume, means the holding capacity of that hemocytometer.
Yes,
Okay.
sir.
Yeah. So that's about the cells. Okay.
Yes, sir.
Yes, sir.
And now maybe like later we will be studying about the rest of the things. Okay. See,
Yes, sir.
if the organism is having only one cell, it is unicellular organism. Okay. Like bacteria, yeast, amoeba are unicellular organism. Okay.
Yes,
And
sir.
Yes,
few
sir.
cells, some of the organisms, just little organisms, they have just 1,000, hundreds to 1,000 cells. We are, multicellular organism. Many, many, many cells are there in our body. Okay,
Yes, sir.
fine. So this is about the cells, all these things. Okay.
Yes, sir.
In
Yes, sir.
next class, we will be studying about the size of the cell, the long, large, all these things we will be studying about in the next class.
Yes,
Okay,
sir.
fine.
Yes, sir.
Thank you, sir.
Okay. Then
Thanks,
take
sir.
care.
Bye, sir.
Bye, sir.
Yeah, see you in the next class. Thank you.